welcome to Edison Open House Global Healthcare 2021. In this session, we're going to be highlighting the work of MaxCyte. It's a global company based in the US, which uses its technology platform to discover, develop, manufacture, and commercialize next-generation cell-based therapies. With me to tell me much more about its work is Doug Doerfler, its president, its CEO, and its founder, all in one. Doug, welcome.
Well, thank you very much. I look forward to this discussion, and I hope you're having a great new year, so thank you.
Now, you've been pioneers in cell engineering based on flow electroporation. Tell us how you've taken that technology and developed it over the years.
So it was originally developed at MaxCyte 20-some years ago, a little over 20 years ago. And we had, my background has always been in non-viral, in viral cell engineering. And I felt that the electroporation system was something that eventually could find its way through and into the clinic and hopefully manufacture medicines for patients. So 20 years ago, we developed the platform with a group of biophysicists and hematologists and cell biologists, and put together what we thought would be the most powerful platform that had the most parameters that we could control to engineer cells in an effective way. And over the course of the last 20 years, we've been able to do it in a format that is completely sterile for clinical use. It has very high performance. More than 90% of the cells that we process are efficiently engineered. More than 90% of the cells are viable. And it's fully scalable. So we can, if a researcher is working at very, very small scale, a drop of cells, they have that same platform that they can use all the way through to treating patients. So it's a very novel technology. We invented it. We've been focused on it solely. And over the course of the last 20 years, we have really been looking at trying to figure out what are the cells that are potentially powerful agents as drugs. And we spend our research and development time figuring out ways of engineering those cells so they can be used for better medicines. So it's an audacious goal, but I also think it's a pretty simple strategy, is focus our technology on making better medicines.
And what type of conditions particularly lend themselves to this cell-based approach?
So what you have to do is, so if I can give a little bit of the biology here, cells are bound together by lipid a bilayer it's called, and it's through electrostatic energy that your cells are kept together. And what we're doing is we're putting an electric field across those cell membranes, so it perturbs them, and then through the same electric field and the ionic strength, we drive the molecule into the cell. It's akin to walking through vertical blinds, where you're the molecule, the vertical blinds are the membrane of the cell you walk through and they close behind you. Well, that's pretty much what we're doing at a submicroscopic level. And we do it with billions of cells through our process. And so sterility is very important, and volume is very important, because you have to have enough cells to make a difference therapeutically, or it's really not worth the trouble of doing engineering.
What kind of medical problems are best helped by this cell-based approach?
Well, it's growing rapidly. Initially, the indications were in oncology, hematologic disorders, blood cancers, and you're seeing a number of leukemias being treated with engineered cells quite effectively, actually. Many of them use viruses. We use non-viral approaches. The next generation in oncology will be using non-viral, because it'll probably be more allogeneic, donor versus autologous, which is a patient's own cells. Additional conditions, and ones we're quite excited about, is inherited genetic diseases. One program that we're working on with CRISPR Therapeutics is a treatment for sickle cell disease. And this is where we take the cells from a patient who has that condition, where their stem cells are producing red cells that are sickled versus normal cells that look more like a frisbee. And we take the cells from the patient and we engineer them using our technology and CRISPR technology, which is a gene editing tool, to correct, if you will, those stem cells. So they go back into the patient and the patient's cells then produce normal red cells. And so those patients no longer have that disease. And we're very hopeful that that will be a long-term effect. So those are the two major ones. We also work in cardiopulmonary disease. We work in infectious diseases. There's been quite a bit of new work that's being done in autoimmune disease and also neurological disease. So I think what you're seeing is cell therapy, although it's a relatively new part of therapies that we're talking about today, there's been 20 or 30 years of work that's been done in this area. And it's finally, as you well know, science takes time. It's finally coming to a point where we can actually apply these techniques and tools to making drugs that can treat, and in some cases, we hope, cure many of these diseases that are intractable. So that really is the mission for, I think, all of us in the field.
So take me through that autologous versus allogeneic, because if I can put it another way, it's kind of off the shelf or bespoke, but actually off the shelf is probably the way that most hospitals would like to see this go. What's your position on that?
Well, we're encouraged by both of them. In the autologous view, where you have a patient's own cells in a more bespoke, if you will, treatment, the whole idea there is you have to make it simpler. You have to make the process faster so that you can treat more patients. And I think that's going to be moving toward a hospital setting at some point in the future. So I think we're going to be, I think, frankly, amazed at how well the technologies are being integrated to do that close to patient treatment. You know, it's akin to transfusion medicine. We don't think about transfusion medicine, but people are apheresis. We're taking red cells and platelets and donating them, right? And so it's the same idea with cells, but in this case, you're adding a step, and the step is you're engineering the cell before it goes back into the patient. Allogeneic is, as you mentioned, off the shelf, and this is coming either from a donor or a group of donors, a pooled group of donors, or a stable cell line. And so that would be more akin to a standard medicine that we pull from the refrigerator and give to the patient. Unfortunately, well, I guess the thing about cells are they're very powerful because they can go to the site of disease, they can sense the disease, and they can react to it and provide the therapeutic effect. And that doesn't happen in biologics. It doesn't happen in small molecules. So it's a very, very powerful technology. On the flip side of that, they're also very powerful, and they can be impacted and impact your immune system. And so just like you do with blood transfusions, you have to be careful that you're matching the donor to the recipient. The same way with cell therapies, we have to be very careful that we're not creating a condition where the patient is rejecting the therapy, which can be just devastating for a patient. So I think there's quite a bit of work that still needs to be done in that area, but we're really encouraged by the companies who are doing this work, many of whom are our partners, and the amount of capital that's coming into it. So I think there'll be a place for both, and it's all about figuring out the best therapy for a particular condition. You say in the UK, I've learned this, different horses for different courses. I think we're going to see that in cell therapy.
So your partnerships are with companies who are working in a particular condition area and then come to you for the specialized cells. Is that right?
They come to us for our technology, which allows them to engineer the cells in their own facility or close to a hospital. So we now have 12 partnerships with most of the leading companies in the cell therapy field, working specifically in inherited diseases. I mentioned CRISPR, but also in immuno-oncology. Companies like, again, CRISPR, Editas, Precision Biomedicine, Allogene, Kite, who has an approved product for leukemia. We're working with all these companies to provide our technology to them so that they can engineer their cells and provide those to patients.
Now, tell me a bit about Karma.
Sure. Well, Karma is our own invention. We identified very early when we've been in this for quite some time that these cell therapies were, we felt that there were some limitations. They've been quite successful in liquid tumors, but in solid tumors, which is the majority, obviously the vast majority of the cancers, they're problematic because of the toxicity associated with these drugs. And the reason for that is that there's antigens on the antigen is the target. That's the protein on a cancer cell, and the antigen on a tumor cell. The cell can't discriminate between a normal cell and a disease cell, and you can manage the on-target off-tumor toxicity, if you will, with blood cancers, but you can't do that with solid tumors because we don't know where these antigens are, and people have died as a result of it. So that's one thing that we recognized, and Karma solves that problem because we use messenger RNA, and mRNA is kind of the top of the fold, if you will, in science today. But it's been a quiet subspecialty in the scientific world, and we use messenger RNA to modify cells. The thing about messenger RNA is that the modification is short term. So that means if you dose a patient and there's an adverse event, the cells are in effect self-limiting. So those cells decrease in their potency over several days, and you can treat the disease without having, hopefully, the toxicity. So that's part of Karma. The other part of Karma is that we can manufacture these cells in one day versus a viral-based therapy that could take a couple of weeks. And that is a very long time for a patient to have to wait for a therapy. So that's the second major advance we're making with Karma. The third is the cost, because you can manufacture it faster, you can reduce the cost. And we really think that some of these therapies are really burdening the healthcare systems. So those three things in combination are what we're working on. It's a very innovative approach. We've taken this program very, very far. We've invested quite a bit of our own capital. We've gotten to a point in solid tumors where the cost of doing clinical trials becomes very, very high, well outside of the ability for MaxCyte to support those. And so what we've decided to do is to suspend our clinical development of this because the costs continue to go up, and find a partner who can then has the infrastructure, understands how to do these very complicated trials, and take that further into the market. The current drug we're developing is for treating patients with ovarian cancer and patients with mesothelioma. So we're quite excited about the technology. We think there's still quite a bit of expertise and capital that's required to make this a real drug, and we're looking forward to finding the right partners that we can work with within Karma.
Do you think there's an issue? I mean, you mentioned it there, that the very high cost of these cell-based therapies, particularly as health systems all around the world are going to be facing a post-pandemic situation in which resources are pretty likely to be very constrained. Do you think that's going to be an issue?
I think it's an issue, absolutely. I think we have to make sure that these cell therapies are being applied to diseases that really can't be treated otherwise. And there is an economic argument to be made, even at £500,000 for a product that will eliminate the need to do a bone marrow transplant, and that's what we're doing in leukemia. And so the cost of the transplant is much higher than the cost of the drug. So in that case, I think it does make economic sense. It also makes economic sense, I think, for some of these inherited genetic diseases where, hemophilia, for instance, and sickle cell disease, where the cost of treating these patients is enormous. And I think healthcare systems, if they look at it holistically, I think they'll recognize the value in these powerful medicines, as they did with biologics, as they did with small molecules. So I think we have to look at this holistically. And really figure out how do we approach these conditions, and how do we figure out how to best treat these patients. But I do agree with you that the Gordian knot here is that the healthcare system, we need to have patients accessible to these drugs, but at the same time they're becoming more complex. And what we focus in at MaxCyte is trying to solve that puzzle and trying to make these more complex therapies easier to administer, easier to produce, and then easier to administer, and easier, obviously, then to commercialize.
Now, you presented some important work at SITC, which, for people who don't know, this is the Society of Immunotherapy and Cancer. Tell us a bit more about that.
Sure, that's around our Karma program, and this was identifying some of the unique characteristics. No one's ever tried to do what we've done using mRNA and using fresh cells. That's why we can do it in a day. And so that presentation and the SITC meeting is a very, very high level meeting with oncologists around really new methods of therapies. And we were highlighted there. We made a presentation, very, very well received, and opened up really a new way of thinking about treating patients with these fresh cells and mRNA. So it was a showcase not only for MaxCyte, but I think in opening up a new area of science that hopefully will encourage other researchers to think more about and devote their careers to. Because I think this is an area of science that's quite exciting, and one that will take a long time, frankly, to create these drugs, but will create, I think, a new class of drugs to treat, probably one of the most difficult areas, the conditions, which are solid tumors, where there really isn't good therapies for many of these, unfortunately, for many of these patients. So whatever we can do to figure that out, I think is a noble cause.
So over the next 6 to 12 months, what should investors look out for?
Sure. So we continue to grow the business quite substantially in the trading update we just had this week. We have given guidance that we will be endeavoring a listing on NASDAQ this year. We're a US-based company, as you mentioned at the very beginning. Many of us have been involved with companies who have listed on NASDAQ, so it's comfortable for us to do that. We also have these 12 partners that I mentioned before. And these 12 partners are all very well financed, and they're moving their drugs through the clinic in sickle cell disease and solid tumors and liquid tumors. And we're going to see those programs move closer to commercialization. And as they move closer to commercialization, MaxCyte will receive milestones upon that progress. And then eventually we'll be able to participate in the economics of those drugs once they're approved. So it's a really exciting time for our company. I think it's an exciting time for cell therapy, and we all have our fingers crossed that the industry can come up with seems to be an effective vaccine and get us back to normal.
And it's worth pointing out that 2020 was a remarkable year in that, it was terrible for many reasons, but 2020 was also the year that we saw the first person with sickle cell effectively cured of their condition. Obviously, that has to be followed up, but it's the first opening of the door, isn't it, into really changing the outlook for a group of patients who have really a pretty awful time of it as with their disease.
Terrible disease, and the patient's name is Victoria Gray, and she's very brave, and she's, as you well know, she's become public about her treatment, and that treatment is enabled by MaxCyte's technology. So we're very excited about helping these patients. It's a devastating disease and very misunderstood, certainly with African Americans in the U.S. obviously, and these patients come in and they present severe pain, and I think there's issues around treating these patients for the pain. They're concerned about potential abuse of drugs, which I think is just very, very horrible position to put these patients in. And so if we can figure out ways of treating these patients and hopefully cure them, and it's a tough word to use because we don't want to create too much hope. There's still a lot of work to do, but it looks very promising, and certainly it's a path that many of us are going to be pursuing with an amazing amount of rigor over the next hopefully several decades.
2020 then was a wonderful high with Victoria Gray, but also was pretty terrible for all of us. How does 2021 look for you and for MaxCyte, Doug?
So you're exactly right, and we have to get through this next stage of the pandemic, and it sounds like we're all making strides to do that, and the sacrifices. I think our company, as many companies, certainly in the life sciences, have adapted using Zoom and other means to communicate with our scientists and patients. So we now have another tool that we can use once we get back to some sense of personal normalcy, that we have this. I think we've been able to have this digital interface now, which I think is going to be something that's never going to go away. We're going to have to really work to enhance it. The future of our company, where we actually have indicated that our growth should increase because of these milestones we're going to receive, and just the progress that we're making in the field of cell therapy. And we believe we enable this field, and we're a vital part of the future of cellular therapies. And we see continued excitement from the investor community in this space. And as that capital comes into our company and other companies in the field, I think that'll set the company up for just extraordinary success in 2021 and beyond. And we just have we're continuing to build a very, very strong team that, again, are focused, as we all are, in making better medicines for patients.
Doug Doerfler, it's been such a pleasure talking to you. Thank you so much.
As well as me. Thank you very much, and be safe.